Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Hello everyone. Today we have a very interesting topic which is using enzymes. It's a very interesting topic and a simple one, but it is a continuation of a topic that we discussed earlier at school, which is enzymes. And as a brainstorming, I did this first slide so as to remind you of the enzymes. So enzymes are chemicals. We call chemicals called catalysts. So catalysts are chemicals. They speed up chemical reactions, okay? And so the enzymes are biological catalysts, and they are biological catalysts, which mean that they made by living things. So the enzymes is the chemicals that speed up the chemical reactions, and we call them a biological catalyst. Okay. Objectives of our lesson today: to understand how enzymes function, and number two, to give some examples of the uses of the enzymes. First of all, enzymes could be used to turn the milk into cheese, and we all know that cheese and milk are proteins. So to make cheese, most of the protein in the milk is turned into solid curd by adding what? Plant juices and animals' stomach juices to the milk. So we add an enzymes, and we all know that, as we discussed, that protease, the enzyme, the type of the enzyme that acting on the protein to convert the protein into amino acid. So we add the enzyme to turn the milk into cheese. Okay, this is one of the uses of the enzymes, to turn the milk into a solid curd, a solid substance, by adding the plant juices and other things. So the curds are then pressed and left to mature. And we all saw this at home. When we want to make cheese, we leave or we put some of the milk and we add some of the cheese or yogurt and press it and leave it to turn into cheese. And we will see this. Protease is the enzymes which convert the milk into solid curd during cheese making. So it converts the protease, which is acting on the proteins, to convert the protein into amino acids, okay, which is the cheese in this case. So making cheese. When the scientists tested plant and animal juices, they discovered what? They found protease enzyme in them. And so the cheese makers have been able to buy pure enzyme made by bacteria. It's not affordable all the time to use the animal juices, but the scientists or in the factories or industries, they did what? They make the enzymes by using bacteria and fungi to make it in a large quantities. So the cheese makers are using what? The enzymes which we can find like a protease to make the cheese. And here we find making sweeter sugar. We have different types of sugar. So we get sugar from fruit, honey, and sugar cane or sugar beet, which is a binger. Most of the sugar in processed food and drinks is made from cornstarch, which is cheaper. But we can use all of these sources, the fruits and honey and sugar cane and sugar beet, but all of them are expensive. So they use this corn starch to make sweeter sugar. Okay. Carbohydrates is the type of the enzyme which act on the carbohydrate to turn the starch into glucose. And as I mentioned before, that starch is the giant molecule. When it's been broken down, it gives us glucose. So the carbohydrates is the enzyme, and we took it from bacteria and fungi are used to break the starch down to make the glucose. We use the carbohydrate to convert the starch into glucose, the smaller or the much simpler molecule. This is a diagram. I took it from the book. Okay, it is going to show us the process of changing the cornstarch from the top into high fructose corn syrup. Okay, we have three main enzymes acting on the cornstarch to convert it. At the beginning, we have at the top cornstarch, okay, and then the digestion using carbohydrates, because the carbohydrates is the vitamin that acting on the starch to convert it into much simpler molecule. We have the carbohydrates from the bacteria, okay. It will give us a molecule with a shorter chain, and as you can see, the molecules in the cornstarch, they are kind of chain, a long chain. Then it's been divided into simpler chains containing four molecules. The 12 turned into 4 molecules. Then a second enzyme is acting, and then the digestion happening again using a carbohydrate enzyme from a fungus or a fungi to give us glucose. And here we can see that the molecule splitting into a much simpler molecules. Okay, so from the beginning we have the cornstarch, one enzyme, the first enzyme, it gives us a molecules with a shorter chain, and then another enzyme, another carbohydrate, it gives us glucose, and this glucose also by a third enzyme, half of the glucose turn into fructose, okay, and the another half of the glucose remains as is it. Okay, the fructose and the other half of the glucose will give us a high fructose corn syrup, which can be used, okay, and it is highly sweet and much cheaper than other sources. A simple diagram, and I hope you can check it again and again from the video or from the book, page 108. Then, as I mentioned, we have a first enzyme and a second enzyme. Both of them are carbohydrates, the first one from bacteria and the other one from fungus. The third enzymes turn half of the glucose into fructose, and that's what we need. And fructose is much sweeter than other sugars, so less needed to be added to make food taste good. Okay, and this is another summary of the diagram. The final results is a high fructose corn syrup. This is the final product, but we started from the top of the pyramid, the cornstarch, and then molecules with a shorter chain, and then glucose. The glucose, a half of the glucose, it gives us fructose, and the other half remains as is it, and this gives us a high fructose corn syrup. Okay, we took till now two examples, one for the cheese, a protein, okay, and the other example for the fructose or for the carbohydrate, okay, and the third example is for fat nutrient, okay. So improving food with lipases. Lipase is the enzyme that acting on fat, and it converts the fat molecule into 3 fatty acids and 1 glycerol. So the lipases make food taste better, especially the cheese. It breaks the fat molecule, as I mentioned, into 3 fatty acids and 1 glycerol. Okay, the fatty acids in the fat molecule is going to determine the properties of the fat molecule and how much they worth. According to the different types of fatty acids, we can determine the properties of each fatty acids. here we have a table for some examples for food and the effect of the lipase treatment. The effect of the lipase treatment on food. Number one, we have the bread. The effect is going to be to last longer and taste better. And then we have the dairy food. The lipase is going to improve the taste and drinks, and the lipases is going to improve the aroma or the smell of the drinks. And then the sauces, it makes it smoother with a smooth texture. And then the meat and fish, fat removal, the effect. Okay, and then finally the health foods, it adds essential fatty acids. So different effects of lipase enzyme on food. So here we can know, or we can see how enzymes work. Enzymes, as I mentioned, the main function of the enzymes or the catalyst is to speed up chemical reactions by making them happen more easily. Without the enzymes, we are going to have a much slower reactions. Okay, so the sugar we get, for example, from sugar cane is sucrose. Its molecules contains two types of sugar, one of them glucose and the other one fructose. So the sucrose from sugarcane contains what? Glucose and fructose, two types. Them for you so as to explain it much clearer. Enzymes shape. The sucrose binds to the part of the enzyme called its active site. Each enzyme has a specific site. We call it an active site. Any type of the enzymes is going to attach to this active site. This is shaped to fit. The active site is shaped to fit the sucrose, okay, molecules and hold it in place. Every enzyme had a different shape, and most enzymes will only speed up one sort of reactions. If we go back to the lesson of the enzymes, if you remember that I mentioned that each type of enzymes is going to work on a specific type of nutrients. So we have the carbohydrates acting on carbohydrate, and we have the protease acting on proteins to turn it into amino acids. And then we have the lipase, which are acting on fat molecules to turn it into fatty acids plus glycerol. So the enzyme is going to act only, or is going to speed up only one sort of reaction. Remember this. Here is an example of the enzyme. Okay, the green circle. Okay, it had at the top the active site, and if you can see the substrate or the nutrient or the molecule, whatever it is, okay, is going to bind, and it fits specifically to the active site of the enzyme. That's why each enzyme is acting on a specific type of molecule. We can see that the substrate is going to bind to the enzyme specifically in the active site, okay? And at the third stage, we have that the product is being converted or digested, and it left the enzyme. Okay, a very clear diagram. Here another two examples of the enzymes, and as you can see at the first one, the substrate, also the shape of the substrate is going to fit the active site of the enzyme, and on the other shape the substrate also going to fit the active site, and it gives us enzyme substrate complex. Substrate is the molecule to be digested or converted. Okay, how enzyme work again. Sucrose needs to react with the water molecules at the beginning to split into glucose and fructose. As we mentioned that sucrose from the sugarcane contains glucose and fructose. Okay, so the enzyme make this reaction much easier. As soon as the reaction has finished, it lets separate glucose and separate fructose molecules go away. As you can see here in the diagram, okay, the first stage we have the substrate entering the active site, assuming this is the sucrose, okay, and the second stage we have the enzyme substrate complex, or the enzyme changes the shape slightly as substrate binds, okay, and in the third stage we have the enzyme product complex, it fits completely or totally, okay, and finally we have the products, two products, okay, leaving the active site of the enzyme after it's being digested or converted. Okay, the enzyme is left unchanged and ready to bind to another sucrose molecule. This is a very important information, that every time the enzyme after the digestion, it's left unchanged as is it, and is ready all the time to bind to another sucrose molecule. And also the enzyme catalyzed reactions are fast, and each enzyme molecules breaks down thousands of sucrose molecules per second. So this is a too much amount, and we need only a small amount of enzyme that can make huge quantities of a chemical reactions, and it's all the time left unchanged. Small amounts of enzymes can make a huge quantities of chemical reacts, and also an enzymes can help build large molecules as well as breaking them down. So not all the time the enzymes help to break the molecules down. It also helps to build large molecule also. And microbes make a huge variety of enzymes. As we mentioned that, we took two examples, the bacteria and the fungus. They make impossible chemical reactions possible and expensive reactions cheap. So enzymes, they are nature's gift to us. They are very important. And this is why our lesson today is very important. More than 3,000 different enzymes have been identified, and the number keeps growing all the time. Thank you for listening. I hope you enjoyed watching the video, okay? And see you next time, inshallah.